During transcription, a single strand of DNA is coded to produce mRNA. The strand is called the template strand.
And the strand that is not transcribed is called a coding strand.
The RNA polymerase will bind to the template strand. It will gradually unzip the double helix by breaking the hydrogen bonds in order to isolate a single template strand.
To form RNA, the polymerase will begin transcribing the template strand corresponding base pairs, replacing thymine with uracil. This process is called elongation.
Once the RNA polymerase reaches the termination signal, it will stop transcribing the DNA and leave. Before the mRNA leaves the nucleus, it needs to be edited. The mRNA consists of sections called exons and introns. Introns are called non-coding RNA, which means that they will not be useful in forming proteins in ribosomes, so that they can be removed from the strand.
To prepare the mRNA to leave the nucleus and ensure its safety, a 5' cap is added to the 5' end of the strand and a poly A tail is added to the opposite end.
Introls are cut out of the strand by enzymes called SNRPs and remain in the nucleus while bonds reform between the exon segments to form a completed mRNA.
Translation starts when mRNA attaches to the ribosome. The initial transfer RNA will occupy the P site of the RNA and it carries a piece of amino acid with it.
The mRNA is also called the anticodon because it brings the complementary nucleotides on the messenger RNA called the codon and ensures the correct sequence of amino acids.
The mRNA passes along the ribosome in short spurts of 3 ribosomes at a time.
Then the RNA is moved to the E site and its amino acid is transferred to the second amino acid, which is just moved to the P site. At the same time, a new codon is presented at the A site.
The initiating anticodon, which now no longer has an amino acid, leaves the E site and the next complementary anticodon comes into the A site.
Each time a new codon sequence moves into the A site, new transfer RNA brings on an amino acid. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. Now the ribosomal complex falls apart by the release factor, then the protein is released into the cell.